based on the monohybrid cross or inheritance of one gene. Mendel proposed two laws, laws of dominance, and another one is the law of segregation or law of purity of gametes. So he obtained same type of results and same type of observation while crossing in other characters also. Not only in the height of the plant, in the position of the flower, color of flower, shape of pod, that is the fruit, he obtained the same result. But when other persons conducted such type of experiments, they are not obtained the same result. That we call as a deviation from Mendelian concept. Some other persons, when conducted such type of experiment, they are not getting the genotypic ratio and the phenotypic ratio are the same. And that type of experiments we call them as deviation from Mendelian concept of dominance. Here are the two examples. 1. Incomplete dominance. 2. Codominance, incomplete dominance and codominance. According to Mendel, in F1 generation, only one character express and other character are not expressed. What is the character expressed in F1 generation? He called it as the dominant, and the character not expressed in the F1 generation, he called it as the recessive. But in case of the incomplete dominance, no parental character expressed in the F1 generation. So in codominance, both parental characters not present. But in case of the codominance, in case of incomplete dominance, no parental character expressed in F1 generation. But in case of the codominance, both parental characters are expressed in F1 generation. So that's why these two comes under the deviation from Mendelian concept of dominance. Deviation from Mendelian concept of dominance. Now first come to this incomplete dominance. Incomplete dominance, partial or mosaic dominance. Incomplete dominance is the phenomenon where F1 generation plant or the hybrids not express any parental character. It is a fine mixture of both parental characters. It first observed in Antirrhinum majus, dog flower or snapdragon. Now first come to the incomplete dominance. Observed in dog flower, also called snapdragon, and the botanical name Antirrhinum majus. Antirrhinum majus. When pure red, flower plant crossed with pure white, in F1 generation we obtain pink instead of red. So F1 generation not consists any parental character. When self-fertilize this F1 generation, possible ways, red, pink, pink, white. So phenotypic ratio 1:2:1. That is red, pink, white. Genotypic ratio same 1:2:1. Genotypic ratio, phenotypic ratio, both are the same. Incomplete dominant, it is the phenomenon of F1 hybrid is the mixture of the characters of the both parents. No parental expressor in F1 generation. It is a fine mixture of both parental characters. And this phenomenon we call as incomplete dominance. It is one of the deviation from the Mendelian concept of dominance. In this one, both parent phenotypic ratio and the genotypic ratio is the same. And another one, that is codominance. Next, codominance. Codominance. Both parental characters are expressed in F1 generation. No one is dominant and no one is the recessive here also. Both characters are the dominant. In case of the incomplete dominance, no parental character is the dominance. But in case of the codominance, both parental characters are the dominant. This type of codominance we observed in both plants and animals. In case of the human beings, one of the example that the blood groups comes under this codominance. In case of the plants, lentil. Botanical name, lens, culinaris, lens culinaris. Here, some seeds, the seed coat with somewhat bigger, large spots, spherical shaped spots. And another trait, that is the small dots. So parent with spots crossed with pure dots. Here just represent here both parents we are representing capital letters. That means both are dominant. That's why we are in this only codominance we are representing the both character, both parents in capital letters. The genotypes of the both parents we represent in capitals because the both characters are dominant. It produces one type of gamete. It also produces one type of gamete. When both they fuse, it's the F1 generation. In this one, large spots, and in this one, small dots. We observe in the F1 generation both large spots and the small dots. So F1 generation express the character of both parents. One is dominant, one is recessive, that is absent. Both are dominant. When we self-fertilize this F1 generation, Here also we obtain phenotypic ratio and the genotypic ratio, that is the same. Spots. Spots plus dots. Dots. 1 is to 2 is to 1. 1 spot, 2 spot plus dot, only 1 dot. Genotypic ratio. Only spot 1. Both spot and dots, that is S and D 2, and only D 1. In codominance also, both phenotypic ratio and genotypic ratio is the same. In case of the Mendelian hybridization monohybrid cross, the phenotypic ratio 3 is to 1. In case of the both incomplete dominance and codominance, the phenotypic ratio and the genotypic ratio is the same. In the first case, that means in incomplete dominance, no parental character expressed in F1 generation. In codominance, both parental characters expressed in F1 generation. No parental character is dominant. Both parental characters are dominant. It's the main difference between incomplete dominance and codominant. Both they come under the deviation from Mendelian concept of dominance. Another one, pleiotropy. If one gene that express more than one character, that phenomenon called pleiotropy.